Welcome back viewers to this channel. This is the intro and the fast forward of genetic engineering or recombinant DNA technology series. From long before we have known that genes are the unit of heredity as it carries all the genetic information to determine the trait of a living organism, and these genes are transmitted from one generation to the next generation. And in today's world, this gene has become the foundation of molecular biological techniques. And as of today, there are many biological and chemical methods to isolate the DNA and identify its base sequences. And one of the methods of isolation and amplification of gene of interest is the cloning of gene of interest by inserting it into another DNA molecule. Now, there are some words that are used specifically in genetic engineering, and they are vectors, recombinant DNA, host cell, and clone. So vectors are DNA molecules that behave as a vehicle and are used for transportation of the gene of interest. And recombinant DNA are DNA molecules that are formed when two DNA molecules of different origin are combined together. And host cell are the cell where this recombinant DNA are inserted and allowed for amplification and replication. And clone, in biology, it is a group of identical organisms that are derived from this single parent. So these are some words that we should know while studying genetic engineering. Now let us understand what is genetic engineering or recombinant DNA technology by understanding DNA cloning. So DNA cloning is the production of large number of identical DNA molecule from this single parent DNA. Suppose this is parent DNA. And these are identical DNA that are obtained from the parent DNA. Now this identical DNA molecule obtained from the parent DNA is known as clone of the parent DNA. Now how do we obtain clone of a parent DNA? For this we need a target DNA. This target DNA will contain the gene of interest that we want to clone. Now, this gene of interest must be inserted inside the host cell. So for this, we need a vehicle. So we also need a vector, since we know that vector is a DNA molecule which will behave as a vehicle and it is used for transportation of the gene of interest. Now, a specific enzyme called restriction enzyme will come and make a cut in target DNA as well as in the vector. Now, after digestion with the restriction enzyme, we will get our gene of interest. And this gene of interest will be ligated in the vector with an enzyme known as ligases. And the vector formed here is known as recombinant vector or recombinant DNA. Now this recombinant DNA will be inserted inside the host cell, and the host cell can be either prokaryotic cell or eukaryotic cell. Let us say we have inserted this recombinant DNA inside the bacterial host cell. Now this bacteria will contain its own chromosome and also the recombinant vector. Now this bacterial host cell will undergo amplification and replication of the target DNA. Now from this bacterial host cell, we can obtain our clone by further analysis or further purification of the host cell. So the whole process where the gene is manipulated to produce a clone of the parent DNA or identical organism or identical DNA from the single parent DNA is known as genetic engineering or recombinant DNA technology.